we're so honored by this invitation to have a chance to interact with this august community of demographers who think about these extremely complicated questions that I do. And I'm a laboratory molecular biologist trained, and I early in my career realized that I couldn't understand the biology of human aging unless I engaged in demographic questions, which is the larger platform that I'll be talking about today. So here is the astonishing breadth of our discussion in longevity, which goes back to great ape ancestor, 6 million years and probably 300,000 generations ago. And if they were anything like the present 4 species of great apes, their life expectancy at birth would have been about 15, and if they reached maturity, it would have been another 10 to 15 years. And currently in captivity under the best circumstances, 55 seems to be the longest they live, maybe 60. So over the next 5-6 million years, species came and went. The genus Homo emerged somewhere 2.3 million years ago, bipedal, tool making, and then anatomically modern humans came into the picture about 250,000 years ago, and there's no reason to think that they would have had particularly great longevity. Of course, we don't have any meaningful demography on human pre-industrial populations until Sweden, and life expectancy there at 30 to 35 to 40 is about the same as it is in various indigenous peoples around the world, Asia and Africa, South America, who are living in pre-industrial circumstances, essentially doubling great ape life expectancies. Life expectancy at birth of 35 to 40. If you get to be an adult, then you have another 30 to 40 years, which means, really interesting point, that almost the youngest child in a family would know at least one grandparent. So fast forward to the industrial age, and in the last 6 to 8 generations, we've added roughly 1 year of life expectancy for every year of historical time. Now we have life expectancies of about 80, and some of my colleagues I think in a gero-euphoric state of mind and expect this to continue as an unbroken trajectory into the future, and I am a little reluctant to accept that, and we can get to that. But this is essentially, in the last 200 years, it's been driven by environmental improvements, principally the reduction of infectious disease and improvement of access to year-round nutrition. And certainly there were heavy genetic things before then, and one that has come to particularly occupy me is the system of gene variants in a major cholesterol transporter, apolipoprotein E, and humans are unique among all species that have been examined in primates in having multiple alleles in our human population. So there are 3 common alleles, ApoE2, 3, and 4. I'll mainly talk about ApoE 3 and 4. And the latest data says that ApoE4 is the ancestral allele and came into human populations 500,000 or 600,000 years ago. The Denisovan lineage has ApoE4. We don't know if it has ApoE3, but it's at least 500,000, 600,000 years old. And ApoE3, by molecular dating, spread in human populations about 200 to 250,000 years ago, roughly at the origins of anatomically modern ape sapiens, and a good 100,000 years before we exited Africa. So I'm going to talk about work from my lab. Here are my wonderful colleagues, Mafalda Pachiatolo, Nick Woodward, Todd Morgan, Hank Chang. So at a molecular structure, ApoE was first recognized as the gene variant contributing the most to differences in blood cholesterol. And the sequencing of it identified that there ApoE 3 and 4 are these substitutions, and the key point is that chimpanzee and mouse have ApoE that looks like ApoE 4 in these two positions, but they have another amino acid here that alters the folding of the protein and gives it a structure that is unique among all ApoEs that have been known. So ApoE4 is evolutionarily novel as a structure in its folding of the peptide chain, and molecular analyses have identified that the arginine or threonine at this position interacts cross-chain to organize how it's folded, and this has a major effect on its membrane interaction with membranes. Quite a lot's known at a biophysical level with that. I don't need to go into this, but the important point is this first narrative that I'll develop is that this influences resistance to infectious disease. And that's not widely known. It's in the literature. So this is a molecule, single peptide chain with an enormous number of effects in different body systems. The neurobiology side of this has received the greatest attention, but it also turns out to influence fat metabolism. So the next thing that was discovered about ApoE alleles is that carrying ApoE4 tended to shorten lifespan, and there's a lot of data on that, but there's an underrepresentation of ApoE4 in centenarians. And various population studies have tend to point, here is the data from Framingham, that ApoE4 carrying women may have greater deficits in longevity than ApoE4 carrying men. So there's a sex ApoE4 interaction that I think is highly important in interest. I'm going to skip that, that's interesting. But so I have been working on Alzheimer's disease from a number of perspectives, and I have to just, as a sidebar, because it's so interesting to me, it may prove to be the case that humans are the only primates with Alzheimer's disease. The great apes and other monkeys have much milder form, and they've never reached the level of neurodegeneration that is in humans. So what are the diseases of aging that are uniquely human is one of the questions that is embedded in the evolution of this gene system. So Alzheimer's disease is women are at greater risk, and the pathological chemistry of Alzheimer's disease shows an excess of the neurofibrillary degeneration and amyloid that characterizes Alzheimer's in women. And we have replicated this in mice, and in fact here is male, female in black, and open bars male, and mice carrying human ApoE have higher levels of amyloid in their brains than male mice or mice carrying ApoE3. So we have replicated this in a mouse, and that is a fairly deep literature at this point that female excess pathology. And there's another side to these sex differences that has to do with spontaneous hemorrhages called microbleeds, and these are now recognized to be a major part of cognitive load that is acquired during aging. And what's fascinating is that here is studies from the US, and there's a comparable data set from Sweden, that in this case there's a male bias for microbleeds in ApoE4 carriers. So we have two different systems with the vascular system and the neuronal system that are proving to have opposite sex biases in the ApoE asymmetry. And to add more complexity to this, in the mouse, the microbleeds have a female balance imbalance rather than a male imbalance. So there's an immense amount of biology that's going to be worked out about this. I just want to bring to your attention that the ApoE story has now a gender asymmetry and at old age, and the question is, does this extend to its effects in younger ages? So ApoE4 is a long list of things that you don't want to know about that ApoE4 makes worse. It's, besides elevated cholesterol, it's pro-inflammatory, Alzheimer's risk. During development, ApoE4 children have thinner parts of the brain in the region that degenerates in Alzheimer's disease. So this interacts with brain development. And recent data shows that the blood-brain barrier becomes leakier in ApoE4 carriers age 30 to 40. And I've just told you that ApoE4 has more cerebral microbleeds. So what could possibly be the advantages of this gene, which is in human populations up to 50%, 45%, and as low as 5%? Well, there are some fascinating data in the literature, analysis of children living in Brazilian shanty towns. Turns out that the ApoE4 carrying children have fewer episodes of diarrhea, and they have better cognitive development in those unhealthy situations. So there's 3 papers in the literature. These are clinical panels, not population samples that would hold up to the scrutiny of our criteria, but they are also supported by some data that I'm associated with in a project with Rudy Westendorf in the University of Leiden, in which he's done a deep analysis of genetics and infections and life history in rural Ghana in villages which differ in their loads of infection. So data that is under review suggests that in this population, ApoE3 carriers, this is Kaplan-Meier, have worse survival than ApoE4 carriers. And comparing villages with low and high infectious load, that there are greater survival of children with ApoE4 than ApoE3. So there's a basis then for thinking that the ApoE4 gene confers pathogen resistance, and this can be tested, is tested here, in mice carrying ApoE3 or ApoE4 in resistance to a gut infection, Cryptosporidia. The ApoE4 carriers eliminate the disease sooner, they regain weight, and in association with higher inflammatory responses, which are part of host defense and important in infectious circumstances. So we now have a platform for seriously considering the relationship of ApoE4 to resistance to disease and infectious disease. And here I want to take us back to the fundamental aspect of our great ape heritage, which is the building of night nests. So night nests are built up in trees, even gorillas do this, protection from predation, and also to get above ground level insects. And they're extremely well designed, and in fact are being considered as an early example of tool use in the choice of branches of various thickness to support weight of large adult. And a lovely article in PNAS on construction of this. But the key point in this discussion is that there are rarely 10% about used more than one night in a row. And great apes do in the morning the same duty that we do and move on to another night nest the next day. So unlike human indigenous populations, they have minimal exposure to their own excreta and that of animals. And here's a photos that I took in the Chimane villages, and I'll come to this in about 10 minutes, which is a typical floor-level culture with domestic animals wandering around. And my 36 hours in this village, you couldn't stay clean. Everything was brown and sticky. You could not keep your hands clean or your face clean. And then consider the living circumstances of people in the northern climates where fuel is extremely hard to come by and water is limited most of the year. There is a level of lack of hygiene that we can't really calibrate. So you figure children in the first 18 months in these confined circumstances. So essentially, our evolutionary history required that we have additional host defense mechanisms that weren't needed by the great apes because of our high population density under close confinement in unhygienic circumstances. We are ship adapted. So, a preliminary map here. Earlier, pre-industrial infections were 90%, 80% of the death. Life expectancy is much shorter. Food was irregular. We now have food in excess of energy needs. Infections were prevalent. Now they're much diminished. And I'm developing the hypothesis that in the indigenous pre-industrial world, ApoE4 was adaptive as part of host resistance, but now it's maladaptive. So there's much to ponder here. You won't find this in the literature yet. It's mentioned in fragmentary form in some of my papers. So I won't tell you about a mouse that we have with the chimpanzee ApoE gene, but it doesn't support neuronal development as well as the human ApoE alleles. Sorry, I won't have a chance to talk about that. So looking into our modern era, I want to sort of set a frame of reference as we go forward with a whole set of environmental issues that have arisen in the 20th century and 21st century that have to guide our thinking in the light of what we're learning about infectious disease and inflammation. So one thing I want to bring out is that our chronic diseases of aging are really the result of a lifelong process. So here is one glimpse of this. The accumulation of lipids on our aorta starts early in life when stillborns analyzed for this showed microscopic foci of macrophages, the phagocytic cell, and oxidized lipids. And clinicians now know that the rate of accumulation of lipids and atherosclerosis is subject to environmental exposure to tobacco. Here at age 25 is the aorta of tobacco smokers versus non-smokers, or coronary arteries. And this is a lifelong process that arises postnatally. And I will bring into this a new source of inflammatory proatherosclerotic processes, which is associated with air pollution. Now, the same processes that are happening in the atherosclerotic lesions, the atheromas, are also present in the senile plaques of Alzheimer's disease. And the same inflammatory proteins and amyloids, cytokines, and inflammatory cells. So this is something that I built a platform of discussion on, but essentially we can begin to ask questions about whole life history inflammatory exposure and how it reads out on chronic processes that have decades long in development, which is absolutely accepted now for the case of atherosclerosis and is under hot discussion in Alzheimer's disease. But they have specific shared biochemical pathways. So, question, what can be said about these as modern diseases? And I think I can document to you that atherosclerosis is an ancient disease, and I want to raise some questions about Alzheimer's disease that wouldn't have had a basis until this line of discussion. So I've been part of a team of cardiologists and radiologists imaging mummies. And they began this 9 years ago, and the top level people who had national status in the American College of Cardiology and Radiology. And they were really interested to know, did people in an earlier time have atherosclerosis or not? And they started on Egyptian mummies, and I was with them in the January 2011 when the Cairo government fell, and that's a whole other story. But essentially, they've demonstrated, I say they because I wasn't doing the hands-on data gathering, but ancient Egyptian mummies have calcification in their arteries in the same places that modern humans do. We got shut down in Egypt, moved to the Americas. There's thousands of mummies in Peru, and we figured out how to analyze them, picked up some additional mummies from the Smithsonian collection that represent pre-Columbian Pueblo dwellers, and some mummies from the Aleutians from Nongan, who were fisher foragers, and the conclusion is that across these populations, they shared the same location and, as close as one can measure, the same prevalence of calcification lesions indicating advanced atherosclerosis. And of course, we can't know the actual ages of these people, and that this is based on bone sutures, and it's uncertain, but we are quite confident that we could identify the fact that in younger adults, in the 20 to 30, we did not find any calcified regions in the arteries, whereas those who were older, 30 to 50, best guess, that was the prevalence of them. So the conclusion then was that atherosclerosis did exist in pre-modern humans, and I'm responsible for this statement in this Lancet paper, which caused a lot of strenuous discussion among our cardiology clinicians, that it is intrinsic in human aging and not linked to any specific diet or lifestyle. So I tell my patients, well, I tell them they're going to die, and I'm not sure whether I can give much advice, but I can tell you not to smoke, to exercise, and not to get too much overweight. Thanks, Doc. So that a really interesting discussion, and an important part of my own cognitive evolution to argue these things out with people who are really smart, working in the silos of clinical medicine, because they see a totally different part of human biology than we do. But it's a very important part of human biology. So at the same time that I was working with this group, I was also privileged to be part of Billy Kaplan and Mike Gervin's project on the Chimane. Eileen Crimmins was with me in this from the beginning, and this is an indigenous population that lives in the Brazilian Amazon, and their demography is essentially the same as Sweden, 1750 to 1770. And that's shifting as part of the package of interviewing by my anthropology colleagues. There's a Bolivian doctor is along, so symptoms are treated and hospitals are arranged for things that need to be done, so their health is improving radically. But the most astonishing thing, and this is really strong, is that heart attacks and strokes are almost unknown. So there's a population, among this 18,000 total population, about 10% of them reach age 60, 65. Their ages are pretty well authenticated from multiple interviews, and I find it to be believable. But the fact is that they don't have heart attacks or strokes as with any definable frequency. And this is completely contrary to expectation from their high load of inflammatory markers that in an ordinary primary care physician's office would say, well, you've got a CRP up there and IL-6, you're in a high risk for having a heart attack. But in fact, their blood lipids are extremely favorable. They have very low cholesterol. This is Chimane versus US. And their blood pressure tends to stay low throughout life. So somewhat interesting statement can be made that inflammation in their lifestyle of a minimal diet, they don't have excess food, and their high work demands does not promote dyslipidemia or hypertension. So I was discussing this with my cardiology colleagues, and they said, I don't believe. And they, on their own nickel, and I was part of this in the last few years, made visits to Bolivia, and here we are going upriver in the dugout, and to these villages, and we brought people back to the regional hospital, whether it was a trade-off or payoff, or however you want to say it, with the local radiologists to get access to the 20-year-old Siemens CT scan that we could. And here is the first group that are being scanned. Here they are. They wear manufactured clothes, and you would think that they're like other folk, but they live in a supernatural world, and it's a totally different set of coordinates. But and they don't speak Spanish, so it's a complex package. But what we found is really remarkable, and that is they do have vascular calcium in the same places, but their rate of accumulation is 20 years slower than North America and Europe. And as a neurobiologist, I said, we've got to get some brain scans. And through an incredibly intricate process, the Albuquerque Human Subjects Board approved scanning brains of Chimane in rural Bolivia. And here's the first data. The number of people who've seen this just went from 10 to a total of 60 people in this audience, roughly. So this is a year, at least before publication. But the point is that we are able to measure gray matter shrinkage with age, and white matter lesions with age, and their rate of change per decade is less than half of what's in the United States. So we're able to ask some really interesting questions. Is slower atherosclerosis associated with slower brain aging, despite the high inflammatory load? So there's a whole set of questions that are being posed at a biomedical mechanistic level that I think have an immensely rich demographic future now that you have a new set of additional variables to bring into this in terms of infectious history. So the past, earlier than the 20th century, was truly in the Darwinian selection for resistance to infection. That was the major cause of mortality. And essentially what we're encountering, and the Chimane encounter, is inflammation that is driven by pathogenic infections, which is highly demanding of energy for fevers and for repair of gut for diarrhea, which also limits food transfer. And then, in our era, our inflammation is totally different in its mechanisms. It's driven by sterile inflammagens, which is a much lower energy demand. No fever, much less repair demanded, and it's a totally different physiology that hasn't been carefully defined, but it's It's known, of course, in the infectious disease literature, but in the sort of larger ecological scale of the current planet, with 4 million people who have chronic infections, their outcomes of aging are likely to be different than in our domain. So here is one schema, and I'm evolving on this. If anybody has comments, I'd love to hear them, but I'm trying to identify the domains of infection-driven inflammation versus sterile inflammation. So let's start with modern sterile inflammation. We have diets that are rich in fat and sugar, which are pro-inflammatory. We're exposed to tobacco, diminishingly, but it's still part of the world. We're exposed to air pollution, which I will get to in the next 5 minutes, which is an astonishing, disturbing story. And our fat depots are, in fact, collections of inflammatory cells. And IL-6 in venous drainage of visceral fat is higher than in the arterial input. So, in our circumstance, ApoE4 and energy excess sterile inflammation is maladaptive, but it could well have been, from the little data that we have, adaptive in the circumstance of high pathogen-driven inflammation. So, opposing changes in the future of human longevity. All sorts of fancy stuff, regenerative medicine, new drugs, personalized medicine, that may be available to a small percent of health elite who can afford it. Anti-longevity trends, which I think are global. I could spend a lot of time on this, but I want to anticipate that there will be an increased load of infections that are part of the global environmental deterioration. Just one example, the rising sea levels are increasing brackish water pools in all coasts, which support insect proliferation and an expansion of insect-borne pathogens. So I think that we will see something reversing our longer-term trends. But I'd like to close on air pollution. And I'm aiming to do this in the next 5 minutes so we have time for discussion. Yeah, okay. I just wanted to get some feedback, because I know you have some cognitive bursts. So, why the heck would a guy who works on neurobiology be dragged into air pollution? Well, I talked to my epidemiology colleagues who have defined in the LA basin a remarkable ecology of disease. And what you see here is the contours of particle density, 2.5 micron particles, which here's UCLA, here's USC, here's Caltech, here's San Bernardino. And essentially across this, longitudinal studies have really nailed down that carotid thickening is greater in proportion to the PM 2.5 micron density in the air. So since the carotid is connected to the brain, I said, okay, I'll do a pilot project, and I've been drawn into something that I never would have anticipated. Too early to tell really how far it's going to go, but it looks like something that is substantive. So a little bit on air pollution. So there's two components that are notable. One is the vapor phase with ozone, oxides of nitrogen, and sulfur. And then there's particulate matter across a size range from sooty particles that you can recognize that are trapped in the upper airways that don't seem to cause much problem. Maybe cough, but not the deeper problems of smaller particles that get deep into the lungs and that also are transported from the roots of our nasal sinuses on our olfactory neurons into our brains. You smell because there's been a transmission of a chemical signal from your nasal passages into your brain. Those neurons transport particles as well. Then the other complexity of this is that there's a wide range of sources of the particles. The bulk of them come from fossil fuel combustion in automotive engines and in coal-powered power plants. Then there's also biomass burning, burning garbage, and then there are earth crust materials that are seasonal from soil, from concrete, and so on. And those all interact to form an extraordinary complexity of particles that differ in their chemistry and physics from morning to noon to night, depending upon the temperature and the ozone exposure and the humidity and the winds. So this is an enormously complex environmental chemistry. So I got, besides the work I was doing in my lab that I'll tell momentarily about, I got my UFC colleagues who I've worked with in the Alzheimer's project to engage in this, and here's a fast flash through of some of the intriguing data. Within the LA Basin, Wendy Mack and Helena Chu, J.C. Chen have been able to resolve different components of air gradients with different age changes in cognitive processes. This is the tip of the iceberg. Across the country, here is Jennifer Ailsher, who's an assistant professor and is doing some remarkable work on this. Across the U.S., extremes of particle density differ by 2 years of cognitive aging. And these are community-based studies. Something of immense complexity is that the same data show SES gradients. You could spend the next 2 hours talking about that. More concretely, white matter changes also show these same relationships. And white matter are the fast conduction pathways that will allow you to, without thinking about it, avoiding what you were going to say to the person you were talking to in a 20 millisecond decision that didn't even reach your laryngeal muscles. It's worth knowing when you're talking to somebody how many things going on before you actually form a word. Often it's best. So here is some data that's under review from the WINS study. First on this side, cognitive decline and all-cause dementia were increased about 70% for those individuals living in zones above the EPA standard of 12.5 micrograms per cubic meter. And there's a hint that there's an ApoE 4 bias here. Standard error bars are too big to fully resolve this. But we, in fact, find the ApoE 4 imbalance in our mice exposed to Los Angeles air pollution. I don't have time to take you through that. So, consistency with the animal models and the human population, it's interesting to put this all in one paper. And we know what points in amyloid metabolism are, and inflammation are being triggered by this. And we have myelin data that goes along with this. And know which synaptic proteins are altered, and we have mechanistic maps. So there's a lot of mechanisms that now we can begin to resolve, and I have to say I was astonished not long after I turned 76 that my NIH colleagues gave me a 5-year R01. So I'm living on borrowed time, but the American taxpayer, I think, will be rewarded. So the last point that I want to bring up, and I'm running over my anticipated time, is that this affects development, and autism is higher in children who are prenatally and postnatally exposed, and childhood obesity also. So here's some remarkable data from Rob McConnell, published this last year, and here's the body mass index for kids who are growing up near a roadway, and here it is in households that have adult tobacco smoke, and here it is adult tobacco smoke and near a roadway. So there's additivity of these, and how could tobacco, what's the common denominator of tobacco particles and fossil fuel particles? Little chips of graphite. And last piece is that we have models that now show this prenatal exposure with responses on high-fat diet. Prenatally exposed have worse glucose tolerance. So I could spend a lot more time on it, but let me just wrap up here by saying we don't know the future of the human lifespan, but it will certainly be environmentally driven, and we may have some insights from ApoE and other inflammatory genes in particular environments. Thank you.